Well, hello everybody. Hope everybody's having a good weekend. I decided to make this little dosary video just to see how this works. I did not get the time in class today to talk about recombinants like I really wanted to, right? We're talking about Thomas Hunt Morgan and his fruit flies and things he discovered with those that were atypical inheritance patterns, right? Mendel didn't find this stuff because Mendel's 8 characteristics he studied with his peas all sort of behaved, the law of segregation and law of independent assortment. But Morgan said, well, yeah, law of independent assortment is true until it's not. And so he first discovered it with sex-linked traits, right? Traits that are linked on the sex chromosome. But he also discovered that these are not just necessarily on sex chromosomes, but any of the autosomes as well, all right? So let's just review and see where we are, make sure we understand this. We'll go step by step, okay? So ordinarily, like with Mendel's study, right, he said, okay, you get one kind of chromosome from one parent. Let's say that's from dad. We'll call chromosome number 1. And let's say over here is chromosome number 7 from your dad. All right, fair enough. Likewise, the homologous pair of number 1 came from your mom, and she gave you a number 7 chromosome as well. We're just picking those out. Now, I don't know where genes are where, but let's just say, for the sake of fun, there's a gene right here on chromosome number 1 that has to do with whether or not you have dimples in your cheeks when you smile. And Dad gave you the dominant dimple gene. All right, and Mom gave you the recessive non-dimple gene, but guess what? Your big D, little d, that's your genotype. You have dimples, okay? But down here, there's another trait. And let's say this one's for attached earlobes. I don't know if it's dominant or not, but let's say it is. Over here are free earlobes, little a for not attached earlobes. Your genotype is big A, little a. You have attached earlobes. You're a heterozygote. Okay? And ordinarily, like the law of segregation says, right, when it comes time for meiosis, big D is going one way and little d is going the other way. That's law of segregation. Right? They are not linked together, 2 different alleles. They're totally separate from each other, and they're inherited separately. Law of independent assortment says that similarly, different genes are not linked either. So just because it seems like a bunch of redheads are blue-eyed, that's just coincidence. Okay? There are redhead, brown-eyed people out there. All right? So dimples would go with attached earlobes in this case. And non-dimples would go with non-attached earlobes. And these would go into gametes, and maybe they'd be sperm. Maybe they'd be eggs. I don't know. All right, but let's say it's a male, and these are sperm. All right? But just as likely, right, is this scenario where here's Dad's number 1 again, but let's say his number 7's over here. A different color, purple. Oh well. And here's Mom's 1 again, and here's her 7 again. All right, but now looking at these traits, this time Dad's dimple gene is on this side, but his attached earlobes are over here now. They moved to the other side of the metaphase plate. Right? Likewise, Mom's no dimples are here, and her unattached earlobe genes went over there. So now, when these separate in meiosis, big D is going to go with little a, and little d is going to go with big A and become a gamete. All 4 of these gametes are equally likely to happen. All right? And that's what's important. So we would say each of these has a 1 in 4 chance of taking place. It just all depends on how they line up on the metaphase plate. That is the law of independent assortment. They assort independently on that metaphase plate. And these are the letters that you put at the top of that Punnett square then. Right? These are your 4 possibilities, and all of them are equally likely to occur. All right, all that's good until these genes are linked. And it just so happens that all of the 8 characteristics of Mendel that he was studying, like flower color and wrinkled peas versus round peas and green peas versus yellow peas and all that, were on different chromosomes. And so they were all just beautifully segregated from each other when it came time to gamete formation, and all of his math worked out beautifully. Well, what if it doesn't? All right, this is what Morgan caught on to. So Morgan said, yeah, sure, okay, well, let's say we'll do a P generation here, parental generation. And this individual is true breed dominant for dimples and attached earlobes. All right. And so on all of his chromosomes, including whatever he would donate to his child, you will see the dimples, and you will see attached earlobes if it's on the same chromosome suddenly. Okay, that's the only gamete that individual can make is big D, big A. That's fine. That would be true if he had several chromosomes, right? The other parent, right, is little d, little a, little a. And they're going to donate a chromosome as well. And we know that little d will be there, and little a will be there. And so this offspring, the F1 generation, they will all be big D, little d, and big A, little a. This is their homologous pair. One parent is dimples, right, with attached. The other parent is no dimples, not attached. All right, sorry, I'm writing with my finger. This is just kind of a mess. Can you read that? No dimples. Okay. And so their offspring has dimples because it's dominant, and it's attached, just like the one parent. Now what Morgan did was he said, just to make things simple actually, I'm going to do a test cross. And a test cross is not where you do a dihybrid cross. You actually do a double recessive. Okay? And so this parent is kind of like this parent. No and not. No dimples and not attached. Okay? And so those are the parents. All right, now we're going to do is we're going to hang with me. What gametes can parent number 1 make for this F2 generation? Okay. And let's put these in here. Well, let's see. Big D, little a. Here's what we're used to writing on these. Little d, big A. Little d, little a. All right. Well, the thing is because his chromosomes look like this. Notice how these are not possible. right? You can't get big D with little a. They went to different gametes. Big D has to go with big A, right there. And little d must go with little a, whichever chromatid it is, right there. But this little d cannot get it with the big A, right? They're on different chromosomes. So I'm sorry, this is getting messy. But the big A is over here, and the little d is over there. They can't be inherited together. These two chromosomes went their separate ways when it came time to make gametes. All right? So this can't happen, and this can't happen. All right? Don't worry about crossover just yet. All right? Assuming no crossover, this chromosome and this chromosome are the only ones that can develop because it's a linked trait. You can't assort independently anymore. All right? Well, the other parent, the only gamete they're going to give you is a little d and a little a. All right? And so you put your combinations in there, and you'll either get big D, little d, big A, little a, or little d, little d, little a, little a. All right, notice how this one is dimples, attach, and this one's no dimples and not attached. All right, so this one represents that parent, just like them, and this one is just like that parent. So these are called the parentals, phenotypes. And if those genes are linked, half of them should be like one parent, and half of them should be like the other parent. What you won't see, okay, is this dimples with non-attached earlobes or no dimples with attached earlobes, because these can't happen. All right? If you get confused by that, rewind this and look at this again. Make sure you get that, because that's really important. All right. All right, now I'm going to have to back this up. All right, should I do it like this? All right, now, rather than just make a new sheet, I'm just going to slowly make this go away to back it up. All right. And what we're going to do now is, what if there is crossover? The only way to get dimples with non-attached earlobes is to cross over. Okay? So let me put these back. Okay. And we'll go ahead and put little d, little a there again. All right. And let's see if I can just kind of do an eraser here. All right. It's like I'm erasing a board. Kind of fun. All right, who's with me? Are you confused? Did you have to stop and rewind? All right. Let's start over now. And we've got the parental units. All right, and there's one chromosome. And let's make it long there. Here's the other chromosome. All right, but these had crossover. All right, so what ordinarily you would have big A and, oops, what just happened? Hold on. All right. Okay. No, wait. Okay. All right. Attached in dimples. But let's say you get a little swap happen here. And these traded places. And suddenly, your attached is on the other chromosome, right? And your unattached, little a, right, which was over here, like paired with that one, got moved. Okay. And this was little d and little d. Oh my goodness. All right, so now all these chromatids, remember, end up in different gametes. And so some gametes are going to look like this first green one, big A, big D. Oops, I kind of got the letters backwards, but it'll look like that. This, yeah, that chromatid will be there. This chromatid will be here. And this chromatid is that one, right? Those are all different gametes. You see that? The big D little a is this big D little a because of crossover. So suddenly these are possible. How about that? Okay. And so we look at big D, big A, little a, big D, little a, little a, little d, big A, little a, and little d, little a. All right. But do these happen in this 1 to 1 to 1 to 1 ratio? All of them equally likely? No. All right, they don't. Because of, what is the likelihood of this crossover happening right in between those two genes? Okay, this is an important concept. The closer A and D are to each other on a chromosome, right? The closer they are to each other, the more likely there's room there for some kind of crossover event. All right, so that would be, let's say you've got a 12% chance of getting a crossover there. Well, what if A was there and D was there? All right, well, in that distance, you may only have like a 5% chance of having a crossover because they're so close. And so this is what Morgan did, right? Let's say he found 800 offspring, fruit flies. They can really reproduce. And around 800 offspring that were just like the parents. Remember the parental, we talked about that, the parental phenotypes. These are the recombinants. Finally, that word I wrote at the very beginning of all this. They've recombined, right? They've got the dimples from one parent and the unattached earlobes of the other parent. And just for the sake of easy math, let's say there's 100 of these and 100 of these. Okay, now here's how you calculate these percentages. You add the recombinants, the total number of recombinants, over the total number of offspring. All right, and so let's see, I got 9. So this would be 200 over 1,800 total offspring, right? Okay, In fact, let's make our math even easier. Let's say instead of 800 offspring here, I got 900 offspring. Okay, so 900 look like one parent, and 900 look like the other parent, and only 100 of each kind of recombinant, give or take. All right. So then my total number of offspring is this nice, clean, wonderful, 2,000. Okay. And so what is my percentage there? All right, cancel everything, and it is, right, 10%. All right. So I have 10% chance of getting a recombinant. He also called these map units, and you'll see that in your reading, a map unit, right? Because the higher this number, the further the distance, like we said, on that chromosome. And using this, Morgan was actually able to figure out how far apart, relatively, genes were from each other that were shared on a chromosome. And he's able to start actually mapping them out, which in certain order. So it's actually the first genomics happened 100 years ago because he was able to figure out how far apart these things were. All right. If these recombinants were more likely, all right, let's say, I don't know, so these were 600, all right, to make my math easy again. So then you get 1,200 total, okay, over 3,000 total, right? So now what's my percentage of recombinants? Okay. All right, in this case, it would be 40%. All right, 40 map units apart. Okay. So what would actually be the highest map units you could possibly get? Right? Well, I say possibly, but if there was no linkage here at all, if they were all independently assorted or crossover was virtually assured, right, then you'd have 900 for every one of these, right, that 1-to-1-to-1 ratio. And then you'd have 1,800 over 3,600. That's 50%. So the highest you can go is actually 50%. 50% means it's not linked. Okay? 50 map units apart means they could be anywhere. They could be on different chromosomes. So it's somewhere between, 1 and 50 or 0 and 50. Okay. And so you've got some problems on there now where you're actually looking at different genes and linked genes and seeing how far apart they are on the chromosome. Okay, and what I'll do for you for putting yourself through watching this video is help you with one of the problems that's on the Chapter 12 study guide. I think it's question number 18, where it's asks you to do recombination frequency for a few different genes. Okay, so let me get rid of this. And so 18 says, all right, you've got a chromosome, and let's just represent one here. Let's say, here's a double copy chromosome. Okay. And you've got 4 different genes on there. So let's just look at one of them, right? A single copy. And we'll just lay it out there. There's our chromosome, or a piece of it. All right. And we're told in the problem that the distance between, like we're looking at J and K, and we figure out recombination frequencies, right? Their map units, or their percentage, is 12%. J and M, man, my handwriting's horrible. J and F are 9% apart. All right, over here I'm going to put K and L are 6%, or 6 map units apart. And L and M are 15 map units apart, 15%. Okay. So this is just like a little fun puzzle. Like, what order are these in? And there's a number of ways you can do this. I like to start with the ones that are furthest apart. This is my MO. So here's L and here's M. And I'm saying they are 15 map units apart. Okay. So let's look at K and L next. Let's look at these two. Okay, so L is 6 from K, so let's say K could be here, or K could be over here. We're not sure. So 6 map units one way or 6 the other. Okay. Over here it says J and M are 9 apart. Well, if this is 15 between L and K here, well then that's 9 there. And J would be there, right on top of, hold on. I'm having trouble with J's. J would be there right on top of K, which is not true, because we're told J and K are 12 apart. Alright, so it's one of them there, or nobody's there. We'll figure that out in a minute. So M is 9% away from J, so I guess J could be over here, right? 9 away, is that possible? Alright. Well, let's find out. So now it looks like this distance right here is 12. And so why not put J there and K there, right? Because they're 12 apart. L and K, yeah, sure enough, they're 6 apart. L and M, they're 15 apart. And M and J, yep, they're 9 apart. All right. And so the order would be K, L, J, or M. All right. In that order on the chromosome. And we have mapped out the order of genes. Now, obviously you could have this backwards, M, J, L, K, right? You could have done it that way too and gotten the same answer. That's fine. The point is, this is the order they are on a chromosome. And so by doing this method and just doing different recombinations of different traits between different fruit flies, Morgan and his students were actually able to start mapping genes out and figuring out what came in what order on certain chromosomes. Of course, they didn't know anything about chromosomes at the time. They didn't even know what DNA really was. It wasn't for another 40 years that Watson and Crick found the whole double helix structure and what chromosomes actually were doing. So they just knew that these genes were somehow linked on some chromosomes and that they were kind of in an order, like on a book or something. All right, so there, I did that one for you. There's another one in there that you can do on your own. If you have any questions, feel free to text me, give me a call. I hope this was really helpful. I apologize for the bad handwriting, but what are you going to do? Okay, I have no stylus, and this is really small. Anyway, hopefully I didn't confuse you. Give me a call if you got questions, and good luck.